Today we're going to be talking about gene cloning. Gene cloning is such a fundamental technique to molecular biology. It's the way that we can get genes into a form that we can use.
To clone a gene, we get the DNA that we're interested in, amplify it, and insert it into a cloning vector, such as a plasmid. The plasmid vector contains the extra sequences needed for the gene to eventually be used in research. The resulting recombinant vector is then replicated in a bacterial cell. We then isolate the recombinant DNA, screen it for our cloned DNA, and it's ready to use.
To clone the DNA that we're interested in this case the insulin gene, obviously first we need to get some of its DNA. It'd be great if we could just get that from the cell.
But because eukaryotic genes like insulin have introns, we can't just extract the DNA. Instead, we extract the cell's mRNA, which has already had the introns removed during transcription. Because we need a DNA version of our gene, we'll use the enzyme reverse transcriptase to make what is called complementary DNA, cDNA, from the RNA. And then we'll make it double-stranded using a DNA polymerase.
Now the repeated cycles are finished, PCR has amplified just the gene that we're interested in this case, the insulin gene. And now that we've got multiple copies of our gene, we need to insert it into a plasmid, which contains all the regulatory sequences that will allow it to be used. And we're going to do this in rather an elegant way.
We cut both the plasmid and either end of our insulin gene with restriction enzymes, and they'll generate the sticky ends that can be used to glue the pieces together. Restriction enzymes recognise specific short DNA sequences called restriction sites and cut the DNA there. This process is called digestion.
Restriction enzymes are naturally used by bacteria to cut up any invading DNA, but we're going to hijack them and use them for science.
To start the process, I have to prepare 2 tubes of restriction enzyme and buffer. I take a sample of some plasmid and I add it to the first tube. Plasmids were also originally found in bacteria and are small replicating circles of DNA that have been engineered to contain a number of different restriction sites inside what is called a multiple cloning site. I then take a sample of the insulin gene that we generated earlier and add it to the second tube. The restriction sites at either end of the insulin gene were added as part of the PCR process. When we designed the primers to amplify the gene, we simply added the restriction site to the 5' end of each primer.
And now, we just leave the restriction enzyme to do its stuff, generating our sticky ends.
When the enzyme cuts both the plasmid and the insulin gene, it leaves overhanging single-stranded ends, which can stick to a complementary sequence.
So now each tube contains the bits that we want, as well as the bits we don't want, like the bits that have been cut away from the end of the gene. And we need to separate them. And the way we do that is using gel electrophoresis. So let's go and do it. Now we've cut our fragments out of the gel, we just need to melt the pieces.
Now that they've melted, we just need to mix the digested plasmid and insert together. Now our insert and plasmid join together via base pairing between the compatible overhanging ends. But that's a relatively weak force.
We need to make a strong phosphodiester bond. And that's catalysed by this enzyme, ligase.
And now we just leave the reaction overnight and let ligase do its thing.
Now that we've incubated this overnight, our ligation should contain some plasmid molecules with the insert in it. In this case, the insulin gene. But we're also going to have some plasmid that hasn't been able to ligate successfully. And we need a way to sort out those bad guys from the good guys. Well, we'll use bacteria. Each bacterial cell will take up one plasmid molecule from our ligation mixture and replicate it so that we can analyse each one separately for the presence of the insert. The process is called transformation, and it's pretty simple. We just mix the DNA, in this case our ligation mix, with some bacteria that have been made permeable. Then we're going to apply a short blast of heat, just to shock the bacteria into taking up the DNA.
Plasmids contain an antibiotic resistance gene and a replication origin. As the bacteria grow, the plasmid replication origin duplicates the plasmid independently of the bacterial chromosome in the host cell. And as the plasmid replicates, the antibiotic resistance gene is expressed. Now the plasmid has had a chance to express its antibiotic resistance gene, I can spread the bacteria out onto a solid nutrient media containing antibiotics.
Then I'll just take this plate to the incubator and let the bacteria grow overnight.
At 37 degrees Celsius, the cells grow and multiply. Plasmid replication is independent of the cell's division, and the plasmids are distributed to each daughter cell as the host cell divides. The number of copies of the plasmid is greatly amplified, with each plasmid producing its own colony of identical cells.
Something's grown. And as it's the plasmid that contains the antibiotic resistance gene, only bacteria that contain the plasmid have grown overnight. So to check which of these colonies contain our plasmid with the insert, we're going to take each colony and grow it in culture overnight.
So first, we need to set up some tubes of nutrient media with antibiotics. And then to each tube, I'm going to add a single colony.
So, now each tube contains a single colony. I'll just take them to the incubator and let them grow overnight. I hope we've given you an appreciation of some of the broad applications of gene cloning. It's such a powerful tool, and it's going to help science to solve some of the problems that are facing humanity. And as future scientists, that's up to you.